Hi, my name is Adam. I am a MCAT tutor and MCAT specialist here at Shemmassian Academic Consulting, and today I'm going to be walking you guys through a biology and biochemistry passage from the MCAT, which is the Medical College Admissions Test. So to start off, where is the biology and biochemistry section? Remember, the MCAT is 4 sections. First we have the chemistry and physics section, 95 minutes. Then we have the CARS section, critical analysis and reasoning section, 90 minutes. And then we get a lunch break for 30 minutes. Then we have our third section, which is the section we're on today, biology and biochemistry, another 95 minutes. And then finally we finish off with the psychology and sociology section to finish out the exam. So I'm walking you through a passage. So there are 10 passages on the biology biochemistry passage and 15 discrete questions, right? 15 questions that are not based off of a passage. So for timing, if you're trying to stay on pace and be able to finish the section within time, we want to aim to finish a biology and biochemistry passage in 8 minutes, right? Because we have 10 passages, 15 discrete questions that we do in 1 minute each. So that takes 15 minutes off the 95 that we have for this section, which gives us 80 remaining, which gives us 8 minutes per passage, right? A good way to practice is if you're going to do just one passage, aim for 8. If you're going to do 3 passages, you aim for 24 or something along those lines. It might also be a good idea for your practice to try to go a little bit faster, maybe aim for 7 or even 6, to give yourself a couple extra minutes at the end of this section or for a more difficult passage to kind of, just give yourself a little bit of breathing room so that you don't have to stress so much and worry about finishing the test on time and being able to answer every question fully. Okay, so let's dive into this passage here. I'm going to be doing a basic passage strategy where you kind of read the passage and highlight it and kind of get a gist for the figures and the paragraphs as to what's going on, and then we'll go over to the questions and we'll try to answer them to the best of our ability. All right, so I'm going to get my highlighter tool out here and I'm going to start here at the first paragraph. Let's zoom in here to the first paragraph here. All right, the heart supplies its own blood flow through the coronary arteries. Okay, well, that's probably what we're going to be talking about. Myocardial infarction, MI. So anytime we see an acronym, we always want to highlight what it means, because that means they're probably going to be referring to it either later in the passage or in the questions. Colloquially referred to as a heart attack, occurs when blood flow to myocardial muscle is stopped and the surrounding tissue dies. Restoration of blood flow after myocardial infarction begins the healing process. Replacement of necrotic tissue inevitably results in remodeling to the structure of the left ventricle, LV, okay? So let's highlight that, which increases stress upon the LV, left ventricle, and may ultimately lead to heart failure, right? So, okay, we're talking about damage, right, Mike? And then we're talking about fixing it, right? So maybe let's highlight remodeling and maybe even necrotic, right? That should be a term that we recognize, meaning death, as we go on here. The study of LV remodeling has been greatly impaired by an inability to observe the process in vivo. Okay, so we're probably going to try and study it. The development of dedicated small animal cardiac magnetic resonance imaging, CMR, okay, so it's basically just MRI of the heart, right? So I'm going to highlight that. Actually, let's add the imaging as well. Has newly enabled high-resolution in vivo assessment of cardiac function and post-MI remodeling in-house models. And remember, in vivo just means in a live specimen, whether it's a human or an animal, right? It's something that's actually alive, right? In vitro would be in a petri dish, or basically just outside of a living organism would be in vitro. So just kind of a little bit more definition of what we're talking about there. Okay, so we're studying it through CMR. That's cool. Post-MI, so let's highlight that for what we're looking at, and mouse models, right? So that's where we're able to do it in vivo. Okay, to explore the efficacy of CMR in monitoring remodeling of the LV wall, 2 commonly used experimental models of MI, so we got permanent ligation, PL, of the left anterior descending coronary artery, right? So that's going to be one way that we cause an MI, a myocardial infarction, would be permanently ligating that LV wall of the left anterior descending coronary, right, the blood supply to the LV wall, the LAD. and temporary blockage, IR, so temporary blockage. So we got two different versions, permanent and temporary, right? Okay, that makes sense. Of the same artery, the LAD, were implemented and assessed. All right, quantities such as the end diastolic volume, EDV, and end systolic volume, ESV, and stroke volume. Okay, we got a whole bunch of acronyms and letters here were assessed. Results are shown in figure 1. Okay, so here's figure 1. Let's look at it, right? So here's end diastolic volume, right? We're looking for something like an asterisk, which would be significance of 0.05, or a pound, which would be a significance between the groups, right? Compared to permanent, right? So we're not looking at compared to control. We're looking compared like the two groups to each other, right? The temporary versus permanent, right? And we notice that we see an asterisk on both of them, and we see a pound, right? So not only are they both significantly different from the control, but they're also significantly different from each other. For both EDV and again ESV, right? We see both signs on the temporary and one sign, so and then no difference in stroke volume, right? You notice we don't see any asterisks or pound signs. So yeah, that's what we want to look for, right? We want to look for significant findings, want to make sure we understand our axes, and then kind of go from there. All right, let's go on. Morphological changes, reduced performance of hypertrophied myocytes, and fibrosis surrounding the region progressively contribute to impaired heart function, right? So we don't necessarily need to understand everything in this sentence to go on forward, but just kind of for your own information, right? We're looking for, or these are like things that happened because we had damage, right? Morphological, I mean, things just look different. Reduced performance of hypertrophied myocytes, right? Just cells, right? Myocytes, muscle cells, myocytes that have gotten bigger, right? Hypertrophied. And fibrosis, right? Meaning we've converted from functioning to more structural, collagen, different types of kind of non-functional cells instead, surrounding the region progressively contribute to impaired heart function, right? So after we have damage, we have these things that make it even worse. All right, the renin-angiotensin-aldosterone system, RAS, right? We should recognize this. This should be part of our outside knowledge, right? We know about this particular hormone system. We also are familiar with a few other axes, including like the HPA axis and the HPG axis and some other hormones as well, right? This should be one of them that we definitely know. And adrenergic nervous systems are implicated in this process. Inhibiting the action of the RAS has been shown to stop or reverse pathological remodeling. So, right, if we inhibit this, then we can stop or reverse. So that's great, right? So we want to do that. So sometimes we want to look to inhibit that. All right. Changes in wall thickness were quantified and calculated based on imaging results, and then okay, so we're looking at this figure is just showing us wall thickness. Okay, so we got the septal wall, right? That's going to be that wall between the left ventricle and the right ventricle, and then the lateral wall, right? That's going to be, because we're talking about left ventricle, that's going to be the wall on the side of our left ventricle. We're going to look at a diagram of the heart in a second, so it can make a little bit more sense. But yeah, so we're looking at this, right? We're going to have no change in the septal wall, right? No asterisks, no pounds. And then in the lateral wall, we have significant change in permanent ligation, and then we still have significance for the non-permanent one, right? And it's significantly different from permanent. So, right, just the lateral, basically only the lateral wall sees changes and the septal wall does not. Okay, so that's going through the passage. So before we move on to our questions, I want to go over some high-yield take-home points from our Shemmassian MCAT content blogs that we have. You can find them at shemmassian.com/MCAT. And I'm going to kind of briefly go through this picture of the heart here from our blog to kind of explain the blood flow. Right, so here we have this figure 2 here, talking about the septal wall and the lateral wall. So where are those? So just briefly before I kind of go through the blood flow, this part right here, this is going to be our septal wall, and I'm going to highlight that in yellow, and then in red, I'm going to highlight that lateral wall, which is going to be running along the side here, right? We can kind of see that there. So these are the two walls that are being described here, right? And remember, the septal wall did not change, right? No change versus control, but the lateral wall changed in both conditions, and they were also different from one another. So as we kind of walk through this diagram here, right, we have our blood flow, right? We have our superior vena cava, right? That's going to be our number one like vein, like all of our veins are coming into the vena cava as we return to the heart. And then first thing that we do is we enter the heart, we enter the right atrium, right? That's going to be our number one. We receive that blood, right? Our diastole, right? Our diastolic pressure is going to be from that. Then we go through our tricuspid valve, right? Our mnemonic for remembering our difference between our tricuspid and our our mitral valve, which also can be called the bicuspid, is try before you buy, right? So you want to try something before you buy something. There's another name for the mitral valve is the bicuspid, right? So the tricuspid happens first, right, on the right atrium to the right ventricle, and then the bicuspid is going to be on the other side. Try before you buy, right? So we go right atrium, down through the tricuspid into the right ventricle, and then that's that first pump, right? We're pumping up into the pulmonary artery, which again, right, this artery is our only deoxygenated artery in our body, right? Our pulmonary artery, our left and right. As we go off to our lungs, we get to our lungs, we get oxygenated, right, through respiration. And then we're going to return back through our pulmonary vein, right? That's right here, pulmonary vein. Right? They're coming in through the back here. See both sides coming in from both of our left and right lungs here into our left atrium, right? That's going to be our third portion of the heart. And then through that bicuspid or mitral valve, right? Because we tried before we bicuspid. And then we enter our left ventricle. That's going to be our fourth portion, which then we get pumped out to the aorta, out to the rest of the body to deliver our oxygenated blood to the rest of our body. So yeah, that's kind of the walkthrough of that. So when we're talking about an MI that happens, we're artificially creating one by permanent ligation or temporary ligation of that artery that provides, our coronary artery that provides blood to this left ventricle. We can see that the damage occurs mostly to this lateral wall because we have significance there, but probably very little to none to that septal wall. Okay, so now that we've walked through that, let's dive into these questions here. So question number one here, let's zoom in on it, get a better look. All right, the cell type investigated in this study are, okay, so right, the cell types we talked about are our cardiac muscle cells, right? We're talking about damage to our cardiac muscle cells. So what are cardiac muscle cells? That's basically what we're asking there. And when it comes to MCAT biology questions, we want to kind of do this game of figuring out what outside knowledge they're referring to with their question and then asking their question very specifically, right? So we're taking the information from the passage, which was that we're talking about cardiac cells and we're talking about the heart in general, and we're applying our outside knowledge of what are cardiac cells, and then we're answering this specific question, which is describing that cardiac muscle cell. So they are striated? Yeah, they are, right? Cardiac muscle cells and our skeletal muscle cells are both striated. We have smooth muscle cells that are not, but this is cardiac, so one is definitely going to be true. Connected by tight junctions? No, they're not, right? They're connected by something called a gap junction, not a tight junction. You can also refer to our blogs if you want a little more information about junctions. And then autorhythmic, right? Cardiac cells are autorhythmic, right? They don't need innervation from the brain in order, like our skeletal muscle cells has to come from our brain, or even our reflexes. Like if you have, you think about our reflexes that don't go to the brain, they still go to the spinal cord, right? They're not autorhythmic. It's still spinal cord and back out, right? So, but our cardiac cells are autorhythmic, right? We have our SA node, which kind of puts down the electricity for our heart to beat by itself without brain innervation. So, right, 1 and 3 are both going to be true, and 2 is false. So the answer therefore must be B. We got 1 and 3 only, consistent with what our outside knowledge said, so that will work out there. All right. Number 2, suppose the resting heart rate of a mouse is approximately 500 beats per minute. Total cardiac output of a healthy mouse is approximately what, right? So we need to remember our formula for this, right? So cardiac output, or CO, is going to be equivalent to heart rate times stroke volume, right? So that's outside knowledge. Again, just just like it's in the first question with MCAT biology questions, we got to be thinking about our outside knowledge with what they're asking us. So heart rate times stroke volume is cardiac output, right? So they gave us heart rate in the question, so that's pretty easy, 500. And now we need to find that stroke volume, right? So let's see here. We probably want to look at this first figure here because that's where we have stroke volume as we highlighted in the end there. What is our stroke volume? So it looks like for all three of them, they're not significant from each other, so we can say that they're all the same number. It's somewhere between 25 and 30. We don't have a precise number, but we can just kind of pick a number between 25 and 30. We might just use 30 for estimation purposes, right? Because remember, this is the MCAT, so we have limited time to do this. So we want to estimate and get as close as we can to the answer, and then kind of round up or down to the actual number when we kind of get close enough, right? So well, let's look at our units first before we figure out exactly what this was. So this was stroke volume in microliters, right? So we got to make sure that we're using our correct units there. All right, so let's look here. Heart rate's 500, right? So let's write that down. We got 500 beats per minute. And our stroke volume we're going to estimate it as 30, right? So let's say 30. And remember, we're going to use micro. And this was just a straight 500, so this would be microliters. Okay, so we multiply those together, right? If we have a hard time with estimation, you can always just multiply the first two numbers and then add the zeros, right? So 5 times 3 is 15. And then how many zeros do we have? 1, 2, 3. So let's add those together. 1, 2, 3. And so our units are going to be 15,000 microliters, right? Okay, so well, we can rule out the 20s right away because those are not close. So it's between the 14s, right? So we got 14 liters or 14 milliliters, right? Remember, micro is going to be 10 to the negative 6th power, right? So, and milliliter here is going to be 10 to the negative 3, right? And so if we want to convert micro into milli, right, we just move our decimal point over 3 places to the left, right? Because our mnemonic for that is, you want to do lad, left you add, or right, this is for moving our decimal point, right r sub. So lad r sub, right? That's our kind of little mnemonic to remember when we're moving our decimal point, which way we go, right? So left, we're going to add. So 1, 2, 3, we're going to add 3 to minus 6, and it becomes to the negative 3rd power, which is what we wanted, milli. So it's going to be C there. Let me get out my yellow highlight tool there and highlight C there. Okay, awesome. Let's move on to number 3 now. Which of the following would result in a decrease of end diastolic volume and end systolic volume when used to treat the mice? Okay, so remember, we have this information from the passage. They told us in this paragraph up here that the renin, angiotensin, aldosterone system and adrenal nervous systems are implicated in this process, and inhibiting that can stop or reverse it, right? So if we have these things change, how do we decrease it so it's back to normal, right? We can look back at these figures again, right? They were increased, they were all increased, and systolic volume and diastolic volume. How do we bring it back down? We need to inhibit, according to this, we need to inhibit the RAAS, or the renin, angiotensin, aldosterone system. So we need to pick some kind of inhibitor. All right, so right away, just looking at it, we see that one's an automatically inhibitor, but is it inhibiting the right thing? So let's see here. So this is called the ACE inhibitor, right? This is, again, outside knowledge of what we should know. This is going to be our angiotensin converting enzyme, right? So that's going to be involved in the RAAS, converting our angiotensin. So that's going to be probably a pretty good answer. Let's make sure the other ones are not true. Angiotensinogen, right, that's part of the RAAS. Angiotensin 2, that's also part of it. So these are both going to be pro, not inhibiting. They're both going to be pro-angiotensin or RAAS system. And then adrenocorticotropic hormone, right, that's going to help us release our different corticosteroids, right, which that's not related to what we're even talking about. That's not going to be related to renin, angiotensin, aldosterone. So totally off base there. They're just kind of throwing in a random hormone. ACE inhibitor, we're inhibiting our angiotensin converting enzyme. That's a perfect answer. It doesn't get much better than that. All right. Number 4, damage to the left ventricle most directly leads to, okay, so right, if I go back to the second page here, right, we got damage to our left ventricle, right, down here, this part of our heart, which, let's remember where that came from, right, the pulmonary veins here, right, the pulmonary veins got the blood from the lungs, the oxygenated blood. It came in through the left atrium, through our bicuspid valve, into our left ventricle, which then pumps out to the aorta, to the body, right? So if we have damage to that left ventricle, we're going to expect everything after that to see some kind of problem. So we want to look for aorta, we want to look for body, we want to look for, something related to that. So let's go back to our question here. Let's evaluate it. So damage to that left ventricle directly leads to A, impaired filling of the pulmonary artery, right? That's going to be our right ventricle, right? Remember back from our picture. Poor systemic circulation, okay, that's looking like in line with what we're looking for, where we're looking for aorta or systemic or body, right? It's looking pretty good for now. C, decreased filling of the left and right atria, right? Unlikely there, right? It's not going to really impact that left atria, right? Because that's right beforehand. If anything, it might be increased on the left because we're not pumping as well out of the left ventricle. So we might even have an increased filling. And then the right atria, maybe. Right? We could maybe have decreased filling of the right atria because we have decreased output from the left ventricle, which remember pumps into the aorta, into the body, into that right atria. So maybe, but not the left, right? So C is not a good answer. And then D, poor performance of the tricuspid valve. Remember, the tricuspid is on our right side, so it's not even on the correct side, and that's going to be between our right atrium and our right ventricle. That's not related to what we're talking about here. So B is our best answer by far. Only one even related. All right, let's look at number 5 here. A lot of words to this one. All right, which of the following correctly demonstrates the sequence of events that occurs after intracellular calcium concentration rises within the cells of the heart? Okay, right, so we want to be thinking here, right, calcium, cells of the heart, right, we're talking about our heart muscle cells, right? We want to think about our cross bridge, our actin-myosin cross bridge formation, which is causing muscle contraction. We want to think about the steps of that. And what calcium is the thing that starts it, right, released from our sarcoplasmic reticulum, right, into causing the beginning of our cross-bridge formation for the muscle contraction. So let's go through this here. So calcium binds to troponin. Yes, it does. So we can start with just that step and see how many of them have that step. B, calcium binds to tropomyosin. It does not bind to tropomyosin. It binds to troponin. Calcium binds to tropomyosin. No. No. So B and D are already out just from the first step. What does calcium bind to? Troponin, right? Troponin is the thing that's blocking those sites on our actin. So it binds to troponin. Troponin then causes a tropomyosin, which is troponin and myosin, right? Conformational change, yeah, that's good. Tropomyosin, so A and C are still both good here. Myosin binds to actin, right? Because now we have space for it to bind on because the troponin got pulled off of it. And then the power stroke occurs, and then ATP is hydrolyzed, or in this case, the power stroke occurs first and then myosin binds to actin. No, the myosin definitely has to bind before, and then ATP is hydrolyzed to help us come back. So A is the only one that makes sense, right? The myosin has to bind to actin before the power stroke. That's how we do the power stroke. Myosin binding to actin and pulling it, making contraction. Okay, that one's good there. And then our final question here, number 6. Pathological remodeling of the heart after a myocardial infarction may lead to decreased blood flow to the lungs. Interesting. Okay, this is most likely to lead to, okay, so when they're just telling us a piece of information, we have decreased blood flow to the lungs. So if we have decreased blood flow to the lungs, well, so just looking at these answer choices before we even try to make some kind of like figuring out what's going to be true, untrue. Metabolic acidosis or metabolic alkalosis, right, is going to be controlled by kind of a combination of our liver and kidneys, mostly our kidney, right? That's how we're going to like filter that blood and make sure that we have the right pH level like out in our systemic circulation metabolic. So both A and B are bad answers because we're not talking about the lungs there. So it's definitely respiratory. So are we respiratory acidosis or respiratory alkalosis, right? So acidosis would mean we're more acidic. Alkalosis would mean we're more basic. So which way are we going here? So if we have decreased blood flow to the lungs, right, that means we're probably going to not be oxygenating our blood in the lungs as much as we want to, which means that our initial blood is probably going to have a little more carbon dioxide than we initially want, right? And we can kind of think back again to our outside information of how does our carbon dioxide and oxygen and pH and how many hydrogen ions are in our blood flow, how does that all relate to each other, right? And we have this thing called the bicarbonate buffer system, right? We can look at that in one of our blogs as well, which is how we regulate our oxygen, carbon dioxide, and how that relates to pH. But the basic gist of it is that if we increase our carbon dioxide in our blood, we also, with that, through the bicarbonate buffer system, increase our amount of H+, which, right, again, if we're increasing H+, that means our pH, which is our measure of how much, our measure of hydrogen ions, is going to go down, right? So remember, more acidic is like more towards 1, 2, 3, more basic or alkaline is going to be more, up to 14, 11, 12, 13, 14. So more hydrogen ions, lower pH, more acidotic, or more acidic. So C is going to be our best answer. More carbon dioxide leads directly to more hydrogen ions, lower pH, acidotic, C. All right, so that's our biobiology passage for today. I hope you guys enjoyed. If you have any questions, feel free to put them in the comments below. And also, if you'd like, you can go into our description beneath the video here, and we have a link there where we can give you a daily MCAT question sent to your email inbox to help you keep up in doing your daily practice for the MCAT, which is important for this process. So thanks for listening, thanks for watching, and I'll see you guys next time.